So as the name suggests, heme agglutination means there will be involvement of RBC. That is why it is heme agglutination. Now agglutination means there will be antigen antibody reaction, but RBC will be here as a crucial point or crucial antigen. That is why it is heme agglutination. Why it is direct? Because directly the RBC is bound by the antibody. That's why it is a direct heme agglutination. Now let's see. So it occurs when RBC acts as a source of an antigen. So here the antigen is, to occur an agglutination reaction, there must be an antigen and antibody. So here the antigen is the RBC, and the serum antibodies, serum from where? That is patient's serum, which contains the antibodies against the particular pathogen for which you are diagnosing, or for any disease you are diagnosing. That antibodies directly agglutinate with RBC and produces a mat-like structure. So what is this mat-like structure? In the test wells. So generally this test can be performed in a slide condition, in a tube condition, in a well condition. So when you are performing this in a well, a mat-like structure will form if there is a proper heme agglutination reaction. At the bottom of the well, there will be a mat-like, clear, round structure. That is the significance, or you can say the test is positive for the particular disease or the particular pathogen. Now this principle of direct heme agglutination test is applied for several diseases, to diagnose several diseases, and named as paulbunnelltest, cold agglutination test. Blood grouping, you all know that A group, B, AB and O, 4 types of blood group can be there, and antiglobulin or Coombs test. So I will be detailing about the Coombs test because it is very important to understand. But in these two cases, if you need a proper class, then write it in the comment section. I will try to have a proper class on this individual tests. Okay. So here, in case of paulbunnell test, CYP-RBC is taken as an antigen, and patient serum antibody is used to diagnose against EBV virus, means Epstein-Barr virus. So for diagnosing Epstein-Barr virus with the help of CYP-RBC, named as or discovered by paulbunnell test. Okay, or the particular name of the test. Now second one is the cold agglutination test. So here human RBC is used to diagnose mycoplasma. So human RBC here is the antigen, and the antibody generated against the mycoplasma in the patient's serum is utilized as the source of antibody, will recognize this RBC and form a agglutination reaction. That is named as cold agglutination because a temperature factor is there. So if I am taking a proper separate class on this, I will detail about this particular test. Now blood grouping, we don't need to elaborate this because it's a very simple thing. And Coombs test, it is a very important aspect of direct heme agglutination test. It is generally applied to human RH incompatibility cases where a RH negative mother bears a RH positive baby. So this mother is pregnant with a RH positive baby. That time a RH incompatibility is there. So let me first tell you what is RH. RH is a factor present on the surface of, or an antigen present on the surface of an RBC. Suppose there is an antigen looking like this. So if this type of antigen is present on the whole surface of the RBC, suppose this is named as A. So the person is, blood group is A, okay, because this present all over the surface of the RBC of the particular individual. Now, apart from this antigen, that A antigen, there is a separate antigen may present in case of several individuals, which may be looking like this. And this is named as Rh antigen. So this is Rh antigen. And Rh, if a person's RBC contains this antigen, then he or she will be A positive. Rh antigen positive means the blood group will be A positive. If there is a and also a separate antigen called B, then it is AB positive. So whenever there is a Rh antigen present on the surface of RBC, that time the person is positive blood group. It may be A, it may be AB, O, or anything. Okay. So separately AB, then O. Okay. If the Rh antigen is not present in the particular individual, that time the person is negative. Negative for what? He or she may be A negative, maybe B negative, maybe AB negative, or maybe O negative. So this kind of factors present, or the antigen present on the surface of the RBC, first discovered in rhesus monkey, that is why it is called Rh antigen. So this is a deciding factor in case of Coombs test. So Coombs test I will be elaborating here. So coming to Coombs test. Now Coombs test can be of two types, direct and indirect. So why it is direct and why that is the indirect, I will tell you. Now first understand, as I told you, that Coombs test is required when you need to diagnose whether a mother is a Rh negative mother, or A negative or B negative or AB negative or O negative mother is carrying either A positive or B positive or AB positive or O positive baby. So when a mother contains Rh positive baby, that blood of the baby will definitely enter into the circulatory system of the mother, and then the mother's immune system will recognize this as a foreign antigen. Which one? That RBC from the baby, as it is a Rh positive. Rh antigen is present on the surface of the RBC present in the baby. So that's why mother's immune system will recognize this as an antigen and produce effective antibody response. So these antibodies which are produced against this Rh positive baby's RBC are nothing but the incomplete or blocking antibodies. Why it is incomplete? Because these antibodies are not totally structured to agglutinate the baby's RBC, at least in the first pregnancy, because it is only blocking the Rh antigen present on the surface of the RBCs from the baby. Now, this is the scenario happens inside the body. Now, that is detected by Coombs reagent. Now, what is a Coombs reagent? One more thing I need to tell you, that blocking antibodies are nature. The antibody's nature is IgG type. So, it is a incomplete IgG type antibody. Now, to diagnose this, we need to add Coombs reagent. Coombs reagent is a IgG type complete antibody which will bind to the Fc portion of the blocking antibody. Adding this Coombs reagent, there will be formation of agglutination, which can be visualized by the technician who is performing the test. Now this test is done in vivo with the fetus serum. Okay, because it is detecting the already bound blocking antibodies are present on the surface of antigen, and then you are adding antiglobulin. Coombs reagent is also called antiglobulin antibody. Antiglobulin antibody. So Coombs reagent as well as antiglobulin antibody is same thing, and blocking antibodies or incomplete antibodies are same thing. But it is an incomplete type IgG and it is a complete IgG antibody which can recognize the Fc lesion of the blocking antibodies, and then the total structure, the RBC bound with the incomplete antibodies, and then this Coombs reagent will form a agglutination process that is visible. So let's understand this by the picture here. So this is the RBC. These green marks are the Rh antigen I have written. Now this Rh antigen are bound by the blocking antibodies. Okay, these blocking antibodies come from the mother's immune system, which now bound to the Rh antigen of the baby. This is baby's RBC, and the Rh antigens are bound by the mother's antibody. Okay, now when you add Coombs reagent, that time what will happen, this antiglobulin or the Coombs reagent, that is the IgG or the antiglobulin, same thing, this will bind to the Fc portion of the, as you can see, the black ones are the incomplete antibodies and the blue ones are the complete IgG antibody or Coombs reagent. So this will bind to the Fc portion. This is the Fc portion of the antibody of the incomplete antibody, and total structure will form an agglutination. So that is why it is a direct Coombs test, because the Coombs test or the Coombs reagent is directly recognizing the fetus RBC bound with the incomplete antibody. It is in vivo. Okay, so that is why it is direct Coombs test. Now what will happen to the indirect Coombs test? Let's see that one. So let's understand the indirect Coombs test. So the name suggests indirect is, here we are not directly recognizing the RBC bound to the incomplete antibodies. We are detecting the incomplete antibodies circulating in the mother's serum. So suppose the mother first pregnancy who delivers a Rh positive baby, she don't know the importance of having a negative blood group and giving a positive blood group baby. So somehow the doctor suggests for the with the case history, the doctor suggests a test, further test as an indirect Coombs test, which will detect whether the mother is having antiglobulin present against the baby's RBC in the mother's serum. So that is detected with the help of indirect Coombs test. So let's see with the picture here. So these are the incomplete antibodies circulating in mother's serum. As we know that immune system will keep the memory response for some time, and this antibodies, incomplete IgG antibodies, will be found in the mother's serum after the first pregnancy or the delivery of the baby. So how it is detected? It is detected in vitro, where mother's serum is taken. Now the reagent here used is the RBC containing Rh antigen. So these are the green ones are the Rh antigen, and this is the RBC, like the previous picture. So you have given this reagent, you have the serum, mother's serum after the delivery, first delivery. Now when you add this reagent, what will happen? The incomplete antibodies will bound to the Rh antigen present on the surface of RBC, as we have seen earlier. Now we have to add the Coombs reagent, which is a complete IgG antibody. anti-human IgG antibody. So this is anti-human IgG body. Now this IgGs will bound to the incomplete IgGs, as I have shown you already in the last picture. So this will give you agglutination reaction. So that is why it is an indirect Coombs test, because it is detecting the circulating antibodies. Which antibodies? Incomplete or the blocking antibodies that is produced during the first pregnancy after and detected after delivery of the baby. So in vitro situation. That is why it is an incomplete Coombs test. So this Coombs test follows the principle of heme agglutination or the direct heme agglutination process. So I hope I am clear with the class. If you have any problem, you can ask me in the Telegram, as well as you can ask me in the comment section. I will try to improvise for you guys more and more. And next class I will be teaching you about indirect heme agglutination. If my classes are really helping you in your studies and it is really clearing your concept, and if you really like my classes, give a big thumbs up and don't forget to subscribe my channel Bio Affairs. This type of subscription and likes give me the enthusiasm to prepare more futuristic classes for you guys.